right, now, there was a man who came up with a different approach. He is, what is his name? Craig Venter. All right, he is the CEO and founder of Celera Genomics, which is a company that deals with genes and genetics supplies. He came up with a different alternative method, okay, called whole genome sequencing. This is the shotgun sequencing that I mentioned earlier. All right, basically what he did was and follow along with me on page 389. He basically took a whole chromosome, chopped it up, and then used computers to sequence it, and then had the computers actually figure out what order they go in. Okay, so and you didn't do any of this mapping, you didn't do frequency of recombination. All you did was just sequence everything and figure out what you got there. All right, and then the computer puts the fragments together. Okay, so he did this, and people did not think that it was very sound, but he did it anyway. And what he got was the whole genome of the influenza virus, all right, the bacteria that causes influenza, called Haemophilus influenzae, all right, it's not a bacteria, it is a bacteria, but it's not the virus, all right, that's what I'm saying. Okay. And then he also sequenced D. melanogaster, which is Drosophila, which is the fruit fly. All right, so should have just said fruit fly in the beginning, but anyway. Okay, so now we figured out ways to sequence whole genomes. All right, we've already sequenced the entire genome for the human. I think we've got the chimp basically done. We've got some other big animals too, but I can't remember what they are right now. So now what? Now when you do with all this sequencing, you've got all this information, all these base pairs in a row. Now what? What do you do with it? Well, it answers a lot of questions for us. Number one, because now we know the organization and control of expression in genes. We know what promoters look like. We know what restriction enzymes are going to cut where. We know the sequence of specific genes, and usually genes that do specific functions or similar functions are located around in the same vicinity on a chromosome. So that's good. We also can figure out growth and development, which we're going to talk about in chapter 21. How genes affect our embryological development, our development past juvenile stage and into adulthood. And then also the big one, the one that everyone cares about, is the fact that now we can compare genomes across species to figure out which parts of our sequences are homologous for evolutionary purposes. Okay, so for evolution, it answers a lot of questions. Where did we come from? How closely related are we to chimps, really? All right, and the answer is like 98.6 or 9% or something. All right, so it allows different organisms' sequences and to be compared, okay, across the board. So we can line up the sequences of C. elegans, Drosophila, the mouse, and us, and figure out where there are homologous parts, where there are completely whacked off, ridiculously different parts, and where there are no parts at all, okay, to see how complex we really are. Now the big kicker, it was kind of depressing to sequence our genome because we thought we are just so much more complex than C. elegans and the single-celled eukaryote Saccharomyces cerevisiae, which is yeast, that of course we had to have probably 100,000 genes, maybe on the big side, all right, whereas the Drosophila fly has like 1,200 or 1,400. So we had to have 1,000,000 times, or we had to have 100 times more genes because we're 100 times more complex. Well, it actually, it was a humbling thing because really we only have about 30 to 40 genes. 30 to 40,000 genes that control all of our growth and development and maintenance throughout our entire life. All right, when you think about how complex we are and when you really think about all the proteins that are needed for everyday metabolic functions, then that really seems very trivial. All right, very few. Why? How then can we be so complex if we only function off of about 10 times more genes than yeast? And the answer is in alternative mRNA splicing, okay, which our old friend alternative RNA splicing. All right, because remember that we have introns, which means that the different combinations of introns and exons are going to give us different polypeptides. And different polypeptides can be arranged in different ways to give us different protein functions. Okay, so when we mix and match the exons and introns in the protein domains, remember the active part that is actually going to do something in the protein, then that's where we get all of our complexity from. All right, remember that it's not just one gene, one enzyme anymore. Okay, it's one gene, many polypeptides, and those polypeptides can be arranged any which way to give us a specific protein. Okay, so now we have a lot of genes in common with yeast and flies. All right, so, and we want to know how, because yeast especially don't have any of the tissues that we have. But how can we have so many genes in common? Well, we try to figure out gene expression in certain tissues and organs, but with DNA microarray assays, right? Or microarray chips is what they would call them. It looks like a little microchip plate, okay, if you've ever seen one in the computer, like on Tron, all right? So it's got wells in it. It's got hundreds of wells in it. And basically you put a microscopic amount of DNA into each one, and then you're going to use your old friend cDNA and hybridize it, and you, when it's being expressed, it'll glow fluorescent because you've added some kind of tag to it, and you'll be able to see which genes are being expressed a lot versus which genes are not being expressed a lot for that specific tissue. All right, and this is on page 20.1, on page 3391, figure 20.14. Okay, and it goes into detail in it. I'm not really going to go that much into detail because I'm not, and I'm pretty sure you'll be able to figure it out. But if you do have questions, just email, call, or text. Okay. All right. So now we've got the sequence. We've got genes. We know about proteins. We know about expression and control. Now we need to figure out what these genes do. All right, because DNA goes to RNA, goes to protein. So what of this DNA is going to make what kind of protein? And how, not what is it going to make, but how does that protein affect the overall function of an organism? Okay, so there's 3 ways that we've learned to test it. Most of the time is going to be process of elimination. Okay, basically you can't possibly be able to figure out all the proteins that are going to do everything in your body and be able to know which one does what and how it affects the development. Because you know that proteins aren't just working by themselves, okay? There can be an amalgamation of tons of proteins that are going to work together as a cohesive unit to make your body work the way it does. So we need to figure out which genes are directly responsible for making those proteins. And more importantly, how the defective gene is going to affect the protein structure and function, and therefore the metabolic process that it's responsible for. Okay, so 3 ways. First one, in vitro mutagenesis. Basically, in a cell, you're going to maybe introduce some kind of chemical mutagen, or expose it to UV light or something. You expose a gene to some sort of mutagen, and then you express the gene in an organism, and you see what happens. All right, so you're doing specific changes to it. All right, the second one is more common in mouse. It's called gene knockout. All right, it's more common in mouse, Drosophila, and C. elegans. It's called gene knockout. Basically, you take whereas in vitro mutagenesis, you're basically changing the specific sequence of a gene and seeing how it affects the protein. In gene knockout, you're wiping out the entire gene. You're basically doing a deletion, and you're taking it out and seeing how it affects the organism now. The third one, and it's kind of complicated, is called RNAi, or RNA interference. And I used to do this a lot with the C. elegans. This is how we kind of generated some of our mutants. You basically expose an organism, during its embryonic phase. You expose an organism to synthetic double-stranded RNA. And they haven't really figured out the entire mechanism for it, but somehow that RNA interferes with regular mRNA transcription and translation and doesn't create a protein. Okay, so when we did it, we were looking at knocking out muscle cell formation, so that a worm could only turn in a right angle. Okay, so it basically went around in a circle to the right. Okay. More importantly, we could also do it to the left. All right, so basically you inject it with with RNA, and you pretty much just wait for, you inject a mom who's pregnant or who's about to be pregnant, or you inject the embryo directly. All right, and then she passes on that RNAi to her embryos, and her embryos intake it. Or you can do this also in vitro as well. You don't have to do it in a live organism, but that's what we did. So you expose the cell to RNAi, and then it interferes somehow. Okay, they haven't worked it out in the book. They haven't really made any progress with it, as far as I know. So I just know that it's double-stranded RNA that interferes with mRNA transcription and translation. Right, that should be enough. Okay, all right. So moving on, we're almost done. Okay, so we've got pretty much a long way still. We've still got like 10 pages left, but this is a long chapter. All right, okay, so where's the future? Where's the future in genomics? Well, we already know so much about genes. We need to start focusing on proteins. All right, and so before I worked in my genetics lab, I worked in a protein lab, and this is what we did. We looked at protein structure and function without knowing anything about the genes before it. All right, so proteomics is the next step. All right, we have genomics, which is the whole set of genes that we look at and how they interact in an organism. Proteomics would be a whole set of proteins, and you look at it, how they help the organism function on a day-to-day basis. Okay. All right, so when we want to look at genetic variation, then amongst these organisms with their genes and their proteins, we can use something called single nucleotide polymorphisms. All right, it's a single base pair variation in a genome, and it happens once every 1,000 base pairs. Okay, so yeah, basically that means that it's a single nucleotide base change. Okay. One every 1,000 base pairs. All right, and it happens throughout your entire genome, and it's what tells me how I'm me, and my sister is similar, but she has different single nucleotide base pair changes that make her unique to her. All right, so when you look at it in the whole scheme of things, 1 out of 1,000 really means that we're about 99.9% all identical. All right, me and the guy who's farming in Cambodia are 99.9% identical, right? Most of the time. All right, it may vary a little bit after that, but so basically we can use those points, they're mutations basically because they're change in the DNA, as markers to figure out how trace back the lineage to when I diverged from the guy in Cambodia. All right, when my line diverged, and how we've evolved since then. Okay, so it's good to have when we talk about evolution to know about that stuff. All right, so make sure you keep it in mind when we go back. Okay, break time. All right, so we're almost done. We're going to talk about applications of DNA technology. I'm going to talk for about 10 more minutes and then we're done. All right, so this stuff is the easy part, I think. All right, how can we apply what we've learned? All right, all these technological thingies and techniques and experimental junk and all these expensive equipment, how do we do it? Well, we can diagnose disease. All right, you know how to look at DNA before it's being expressed, while it's being expressed, after it's being expressed, when it's not being expressed correctly. So we can diagnose disease before birth, all right, while you're still in your mama's womb, we can figure out what's wrong with you. They call it an amniocentesis. We can look at pathogens like HIV because we've sequenced the DNA of that virus, so we know exactly what we're looking for. If maybe you can't see it in the blood, you can use a probe to figure out if there's any sequences that match that, so that a person who doesn't know that they have HIV can be confirmed as having it or not having it. We can ID carriers. Okay, for those recessive mutations that you have to have 2 copies and they show up and people are like, what? How can I have this? No one in my family's ever had this. All right, then you can be able to figure out who the carrier is. All right, who's the one who's contributing half of the problem.